This presentation discusses about Hodgkin's lymphoma. Lymphoid neoplasms are of two types, leukemias and lymphomas. Lymphomas are tumors that produce masses in lymph nodes or other tissues. There are two groups of lymphomas, Hodgkin lymphoma and non-Hodgkin lymphoma. Hodgkin lymphoma was first described in 1832 by Thomas Hodgkin. Being considered as one of the two main types of malignant lymphomas, it is still a potentially curable disease. In recent times, they have been understood to be an unusual tumor of B-cell origin. They are distinguished from non-Hodgkin's lymphoma by their unusual pathologic and clinical features. Hodgkin's lymphoma can be defined as a distinctive group of neoplasms that are characterized by the presence of a tumor giant cell called the Reed-Sternberg cell. They may arise in a single lymph node or a chain of lymph nodes. Then they spread in a stepwise fashion to the adjoining lymph nodes. The causative agent of Hodgkin disease is unknown, but they are believed to be associated with genetic and environmental factors. Infectious agents such as Epstein-Barr virus may be involved in the pathogenesis. In as many as 50% of Hodgkin disease cases, the tumor cells are EBV positive, and almost 100% of HIV-associated Hodgkin disease cases are EBV positive. Risk factors. Number 1. Patients with HIV infection have a higher incidence of Hodgkin's disease compared to a population without HIV infection, but Hodgkin's disease is not considered as an AIDS-defining neoplasm. Number 2. Genetic predisposition may also lead to Hodgkin's disease. Number 3. Siblings of an affected individual have a 3 to 7-fold increased risk of developing Hodgkin's disease. This risk is higher in monozygotic twins. Characteristic features of Reed-Sternberg cells. These are large cells of 20 to 50 micrometer in diameter. They have an abundant amphiphilic finely granular cytoplasm and two mirror image nuclei, with each nuclei having an eosinophilic nucleolus and thick nuclear membrane. The two mirror image nuclei resemble owl eyes. Chromatin is also distributed at the cell periphery. Hodgkin's disease can be histologically classified into 5 subtypes: nodular sclerosis, mixed cellularity, lymphocyte depleted, lymphocyte rich, and nodular lymphocyte predominant Hodgkin's disease. Nodular sclerosis type comprises 60 to 80% of all cases, and it shows a nodular pattern. The characteristic lacunar type Reed-Sternberg cell with mono or multilobated nucleus and small nucleolus with abundant and pale cytoplasm is seen here. Mixed cellularity type comprises 15 to 30% of all cases. Reed-Sternberg cells are large with bilobate double or multiple nuclei and a large eosinophilic inclusion-like nucleus. Lymphocyte depleted Hodgkin's disease makes up to less than 1% of all cases. Here, large number of Reed-Sternberg cells and bizarre sarcomatous variants are present with a diffuse and hypocellular infiltrate. Lymphocyte-rich type comprises 5% of all cases. The classic or lacunar type of Reed-Sternberg cells are seen here. Clinically, they are similar to the mixed cellularity type. Nodular lymphocyte predominant type comprises 5% of all cases. Unlike other subtypes, here the Reed-Sternberg cells are observed rarely. Instead, a variant of the Reed-Sternberg cells, called as the lymphocytic and histiocytic cells or popcorn cells, are seen here. They are called so because their nuclei resemble an exploded kernel of corn. Ann-Arbor staging classification for Hodgkin's and non-Hodgkin's lymphoma. According to this classification, lymphomas are categorized into 4 clinical stages. Stage 1 denotes involvement of single lymph node region or lymph node structure. Stage 2 denotes involvement of 2 or more lymph node regions on the same side of the diaphragm. Stage 3 denotes involvement of lymph node regions on both sides of the diaphragm. Stage 4 denotes disseminated involvement of one or more extra lymphatic organs with or without associated lymph node involvement, or isolated extra lymphatic organ involvement with distant nodal involvement.